This is our Dirty DNA Extraction Science Communication by Cole, Abby, Hia, Jason, and Aaron. And we're from Tuesday, Thursday Biotech, and we're group 4.
Our purpose is to inform middle schoolers and 6th graders who are just getting into life sciences about the foundations of cells and DNA. We want them to understand and at the same time, and at the end of this presentation where DNA is in a cell, what it does for the human body and how they can extract DNA from plants at home. We also want to focus on the different aspects of a lab and what a controlled experiment is and how they can conduct one too. And to engage the audience, we will have them ask questions throughout the presentation and ask them to perform the experiment themselves at the end.
So before we discuss how to do this lab, we have to look at a bit of cell biology. Now, our body is composed of trillions of cells, and a cell is the smallest unit of life, and it's the building block of all living things. Now, cells work with other cells to make tissues, which then make organs, and which then make organ systems, which help run your body. And all of these work together to ensure that you survive. But cells also need to operate properly to do this, so they have organelles that have different tasks to do, which essentially means that they have different functions to perform. Now, one specific organelle, the nucleus, contains DNA, which is genetic information, and it lets you inherit genes such as eye color, hair color, and such from your genetic line, which are your ancestors. And as you can see where the arrow is pointing the diagram, that's the nucleus, which contains the DNA, which is what we are going to do in this lab to extract it.
The goal of this lab is to extract DNA from a plant cell, in this case strawberries or bananas. The materials that we need for this lab are salt, dish soap, cold rubbing alcohol, the fruit we chose, a stirring rod, glass, Ziploc bag, paper napkins, and measuring spoons. The purpose of this experiment is to perform a controlled experiment. In an experiment, you perform multiple trials to have more accurate results. In a controlled experiment, you only change one variable each trial so you can control it. In this case, the controlled variable is salt, and only change the amount of salt used and see which amount is best to extract DNA. We will use rubbing alcohol to help the DNA precipitate or rise to the top of the glass so it can be extracted. Baking soda to help lyse or break open the cells. It has chemical properties that will open up the cells and release genetic material, which contains the DNA we want. Salt helps to make sure the cells do not break when you mush the fruit up. It keeps the DNA intact so that you can extract it.
And the procedures in order to extract the DNA. So first, we're going to take a half a cup of strawberries or 1/3 of a banana and put it in a Ziploc bag. Once it's in the bag, you're going to add a small amount of water and 1 teaspoon of salt to the bag, and you're going to close it and then smush those substances up. In a separate glass, add 1/2 cup of water and 1 teaspoon of baking soda and mix the solution. Once you've mixed the solution, you're going to add the strawberries or bananas that you mushed up to the solution and stir for 15 minutes. After you're done stirring, strain the solution using a coffee filter or colander into a separate glass. Once you've strained the solution, add cold rubbing alcohol and your specified amount of salt to the solution because each group member has a different amount of salt that they were assigned. Wait for 5 minutes after adding the salt and rubbing alcohol until you see a thin white colored layer rise to the top of the glass. That white layer is the DNA, and in order to extract it, use a stirring rod or a toothpick and gently scoop it up and put it down on a paper napkin. And the final step to extracting the DNA is to measure the amount of DNA that you got from the solution.
What were the outcomes of our experiment? We have determined that the optimal amount of salt was 1 teaspoon as it extracted the most DNA. What does this mean? If we look back at our experiment, DNA has a backbone with negative charges. The chemical properties of salt cancel these charges and bind the DNA together. But too much salt means that the chemical properties would be too strong to perform the job. This also applies if there's too little salt, but the chemical properties would be weakened instead of strong. The best amount of salt to use is 1 teaspoon because it'll extract the most amount of DNA due to its chemical properties. Now it's your turn. You can try this experiment at home by controlling another variable, such as the amount of dish soap. Some safety reminders is to practice lab safety by incorporating basic hygiene like washing hands and having a clean surface. You should also try documenting your work in a notebook so you can repeat the experiment again.